Welcome back, Dr. McDonald here, and we're back into eukaryotic cells, and now we are going to talk about the eukaryotic cellular biology. So what we're really going to be talking about is a lot about proteins. So hang on to your hats and let's get started. Well, cellular proteins, they are the workhorses of the cell, and so we've got to start off with what is it that they're doing. Well, first of all, let's remember that they're a macromolecule that is actually a polymer of amino acids. And these amino acids are connected by peptide bonds, strong covalent bonds called, and therefore, since there's a lot of them, they're called polypeptides. Now, different proteins are going to have different sequences of the amino acids, and this is very important. They're going to fold differently as a result. In fact, as the sentence says there, shape is everything in the world of proteins. If the protein does not assume its proper shape, it cannot function properly. And when that happens, the cell actually has mechanisms for recycling the proteins. But after a while, sometimes they're not capable of doing that. And that's when disease happens. Now, some proteins are also modified after synthesis. So this was referred to because protein synthesis is also known as translation. This is known as post-translational modification. And this is going to alter, again, the protein shape and function. Now, sometimes it's supposed to do that, other times it's not to. And as I've already said, altered protein function leads to disease, whether it's caused by a genetic mutation that you inherited, or from a spontaneous mutation, or because of aging. Now, when we want to study all of the proteins that are being made in a particular cell at a particular time or under certain environments, this is known as proteomics. And we're looking at all of the proteins that a cell is providing, say, under normal conditions and then under different abnormal conditions. We're going to still talk about their functions, but first off, we're going to start off with membrane proteins. And as you recall, these can be either transmembrane, integral, in which case they extend across both layers of the lipid bilayer. And of course, those that do are going to have important roles in the transport of molecules in and out of the cells, and something we call signal transduction, which is going to take the information that the cell receptor has bound something and then turn it into a signal to the nucleus that will cause a change in cell behavior. Now, the type of membrane proteins present depends on the function of the cell. You're going to find different transmembrane proteins in a kidney cell than a liver cell or a cardiac myocyte. And if we're talking about peripheral proteins, however, we're not going to be talking about transport so much. They'll have different functions, but they'll be just in one layer of the bilayer. So they'll be at the surface or inside the cell. Now, membrane proteins have a lot of important functions. They are going to be synthesized on the ribosomes located, if you recall, on the endoplasmic reticulum. They'll then be transported through the Golgi to different membranes throughout the cell. So it could be the plasma membrane, it could be membrane of a lysosome, or another one of the membrane-bound organelles. Is this important? Yes. There's some very serious, sometimes fatal genetic diseases that occur when proteins don't get to where they're supposed to be going. Well, what do membrane proteins do then? Well, they can serve as cell surface markers. That's good for us in terms of identification. Sometimes they have other function, of course, for the cell. They can have cell adhesion markers, so they can adhere to the cell next to them or to the extracellular matrix. They can function as catalysts, so they can be enzymes. Again, they can be transport channels. Active pumps, meaning they need energy to transport, say, the sodium and potassium and calcium ion pumps. And then energy transduction and ATP production. All of these we're going to be talking about this week. So here's just a picture of the different functions that a membrane protein can have, particularly a plasma membrane protein. Here you can see where on the lower right here, attachment to the cytoskeleton, and then also attachment to the extracellular matrix. outside of the cell, so you can see how these membrane proteins are very, very important for the cell. A cell is a living organism, but unlike multicellular organisms such as humans, it doesn't have a sense of smell. It can't see things, it can't hear things, it can't taste things, but it can touch things, and it can touch things through proteins known as cellular receptors. Now, cellular receptors are found on cell surfaces, but also within the cytoplasm or the nucleus of a cell. Their function is to bind to a specific molecule. The general term is ligand. It's also also pronounced as ligand. An example here is the epithelial growth factor receptor, EGFR, and epithelial growth factor, EGH. I should say growth hormone. And then you have estrogen receptor and estrogen. So this is one example of receptor-ligand interaction, which is involving hormone interactions. Well, the signaling cell is going to make the ligand. The target cell will have the receptor then that binds it and then responds. So binding is just one portion of what happens when to a cell when the receptor binds a ligand. Now, how the cell responds then after a receptor has bound a ligand, it depends on the different signal transduction pathways that are in the cell. Now, these signal transduction pathways are taking the signal that a receptor bound its ligand, transducing it into a change in cellular behavior. So it's got to go through a number of sort of pathways. I always think of it sort of as a domino effect. So the receptor starts with binding in something, and then the dominoes start to fall down until it reaches the nucleus and turns on and off different genes. When that happens, the response to the cell could be that it will proliferate and make more cells. It will stop growing, or it will increase in size, or it will die. This is what happens after a receptor binds a ligand in the cellular world. Receptors are classified on the basis of their location and function. They determine the ligands or the hormones that a cell can bind and therefore respond to. And the ability of a hormone or a ligand to stimulate a cell is regulated by how many of the receptors are present in or on the cell. So if you take a look at this picture here below, you see in upregulation you have more receptors on the target cells, so that makes it more sensitive. So you can have lower levels of the hormone present and the cell will be able to respond because it's got so many receptors on its surface. However, if you got too much hormone around it, this is what happens with drug addicts. If they take too much of a particular drug, the cell is hypersensitive, so it will downregulate the receptors, and you're going to have more of a substance in order to activate the cell. So how many receptors are on a cell is going to influence whether it can respond to lower or higher concentrations of the ligand. So let's go into this concept a little bit more, because students can, and people just in general can get kind of confused with this whole receptor-ligand interaction anything. Anyway, again, a ligand or a ligand is a general term for any kind of molecule that binds to another molecule. What can ligands be? They can be steroid hormones, neurotransmitters, drugs. That's what we typically think. But they can also be oxygen and carbon dioxide. You also have to think of them as being antigens or allergens, such as pollens, how about pollens, that will bind to lymphocytes, and we'll be talking more about antigens and allergens when we get into the immune system. A receptor then is going to be a protein, and part of it, of course, is going to bind that ligand. But then it's got to somehow generate a cellular response. So part of it is also going to probably undergo a shape change to begin that domino effect that I mentioned earlier, that occurs when a receptor has bound its ligand. So the shape of the receptor is going to change when it binds it, but the shape initially is going to determine which ligand the receptor can bind, because only one that fits in the receptor, the binding portion, will be able to activate the receptor and then the cell. So we have a picture of it in this slide. If you take a look up here, we've got a ligand, and of course it's going to fit very well. This looks kind of like a tool, if you will. And then it binds to this, and what will happen is, as a result of binding, this will alter its shape. And so, say the signal transducer, one of the first things, can bind to it, and then you get this domino effect. So the generic term for things that bind to a receptor is ligands or ligands. And though ligands can bind to receptors, causing a change, they also bind to lots of other molecules that will not affect the cell. And lastly, receptors can be found in cellular membranes and within the cytoplasm of a cell. So just keep that. Everybody, I know when we first learn about it, they're on the surface of the cell, but they can also be inside and even in the nucleus as well. There are three major classes of receptors. There's what we call an ion-linked channel, and they will bind a ligand, and as a result of this, certain ions are then able to cross the membrane and enter or exit the cell. So the ion-linked channels will open and close when they bind their ligand. Enzyme-linked receptors are going to be a little bit different. Typically, they are what we call protein kinases. And a protein kinase is just an enzyme that will add a phosphate group to another one. And so they mediate the transfer of phosphate groups, say from ATP or GTP, to proteins, which are then going to affect their function. So this is frequently the start of one of those domino effects that is going to end up into a change in the cell behavior. You also have G protein linked receptors, okay? And these will act indirectly. Again, there's going to be a membrane protein that when it binds something, it's then going to lead to the splitting up of GTP. Now that's guanosine triphosphate. It's an energy molecule, and what it does is it's going to turn another protein on and off. Now, G proteins usually activate certain target enzymes or ion channels. So this is, again, it's one thing happens, then the next thing, then the next thing. Again, it's this domino effect. They are going to target enzymes that generate something that we call second messengers. So if you think of the ligand as the first messenger, and think of this as a Pony Express thing, and it binds to the first receptor, and then that first receptor is going to activate a second messenger, and they're going to then ride it to the nucleus so the cell can change its behavior. And there are three different types of G protein linked signaling pathways. GS is stimulatory, GI is inhibitory, and I forget what GQ stands for, so you don't have to remember it. But it's something that's kind of in between. It can be one or the other. And there are actually a lot more classes of receptors, so be grateful you're just going to have to learn about the first three. But these are really very common, and you will hear about them, and there are genetic disorders and stuff as a result of mutations in these receptors, and they can have rather far-reaching effects depending on the cell type. Let's take a look at these receptors. If you take a look at the one on the far left, this is an ion channel linked receptor, so the ligand binds and now the channel opens and the ion can come through. Or it can be closed as well. Here we have an enzyme linked receptor, so the ligand binds and now it activates this receptor. It's going to frequently take a phosphate off of this and phosphorylates itself and then phosphorylate other enzymes. And our G protein coupled receptor, as you can see, spans the membrane a number of times, but it will have its receptor portion here where it binds the ligand, and then it will interact with that G protein, which will then activate an enzyme which generates the second messenger in the Pony Express. This is showing you more of the G protein coupled receptors, whether we have the stimulatory pathway on the top or the inhibitory on the bottom, and this GQ activates kind of a number of them, so which we're not really going to go into in that great of detail. Let's look at the process of signal transduction a little bit closer. It begins when a receptor binds its ligand, and this will induce a conformational change in the receptor. The altered receptor shape inside will then trigger these cascades or domino effects that I've talked about. It's going to generate second messengers, and the two most common second messengers are going to be something called cyclic AMP, CAMP, and calcium ions, or Ca2+. This will then lead eventually to activating proteins in the nucleus, which will affect transcription of proteins or the replication of the cell. And by transcription, what we mean is that the gene in the DNA will be transcribed into messenger RNA, and that's a transcription, and then translated into a protein. So when a receptor binds a ligand either from outside the cell, and they can bind it on the cell surface or inside the cell, what's going to happen is there's going to be a change in the proteins that are made in the cell, and that will allow the cell to change or respond to that ligand. So again, it's leading to a change in cell behavior or activity. The second messengers and enzyme cascades are what takes the binding of the receptor to the ligand to the nucleus, as it says there. Along the way, the cascades will amplify the signal, so it will actually cause a change in the cell behavior, and distribute that signal to other pathways, so you can get complex intermediate responses, etc. And you can modulate it when other substances bind to other receptors. So it's a very complex process within the cell. This figure is showing you just what signal transduction is going to do. So let's take a look at the upper right here. Here we have a receptor. It binds the ligand, and then it's going to relay that information, amplify it through these second messengers, so it gets to the nucleus and change gene expression or regulation of the metabolic pathway changes in the cytoskeleton. So it's going to cause a change in behavior. You see the same thing on the upper left here. Again, an extracellular signal, and then it's going to transduce the signal to the nucleus, and in some cases cause it to differentiate, sometimes cause it to divide, sometimes cause it to die. And this is, again, just a figure to show you, again, the sort of the different types of things that can happen. So here we have a messenger binding, and sodium now comes in. And this alters the electrical activity of the cell, and this is important, of course, in generating action potential in muscle cells. And then you've got, again, another messenger binding receptor. It's going to associate with a G protein, and now this will activate, say, a calcium channel. So as you can see, these are the different mechanisms by which these ion channels can get activated. Okay. Now we need to talk a little bit more about these second messengers. So a lot of these ligands, they'll bind to the cell surface receptor, but they're not entering the cell. So they've got to, it's a relay. They've got to pass on the signal, and they're going to pass it on to the second messenger. Now, the second messengers, you don't just have one. They're generated in large numbers. They diffuse away from the cell signal, and they're going to spread the signal throughout the cell. And this is what you want them to do. As I've already mentioned earlier, there are two major second messenger pathways. There's the cyclic adenosine monophosphate, or cyclic AMP, CAMP pathway, and then the calcium ion, or Ca2+. That's a calcium ion pathway. Those two pathways are demonstrated on this figure. If you take a look on the left, you see the first messenger is binding the receptor. It's a G protein, and it's going to activate adenylyl cyclase, which is an enzyme that produces cyclic AMP. And this then binds to other enzymes, activating them, bringing about a change in the cell behavior. On the right then, we see binding of it, and in this case, it's going to activate another G protein, but it's going to result in the activation of phospholipase C, and this activates, actually breaks down a lipid in the membrane, but what it's going to do is generate these two molecules. Don't worry about them, but it's going to lead to an increase in intracellular calcium, and that increase in calcium then activates various proteins, which then lead again to a change in the cell behavior. Now, you say to yourself, and this is, again, believe it or not, a simplified picture of what really is going on. So don't worry about it, but you will occasionally hear about all these different things like RAS and maybe PKC and things like TAC or some ERK or something like that. And what they're talking about is this communication pathways within the cell. And when these get messed up, sometimes you keep the cell cycling. Sometimes you kill the cell. And this sort of thing. And this is what can lead in cases where it's cycling into cancer and other things. So you can see why this has got to be a very complex system within the cell. Because this is the cell's, remember, only sense, a sense of touch through the binding of receptors. So it can respond to changes in its environment. Now let's step back from the inside of the cell and see how two cells are going to communicate with each other. There are three mechanisms by which they can do this. They can use gap junctions, which we don't see on that many, but on a very important cell type. Contact dependent is very important, particularly with epithelial cells and other cells that line surfaces or entrances. And secretion, which is the most common, and you will see the different types of secretion that can occur. And finally, something that's not mentioned in your textbook, but I think is important because it's something that does occur between cells and something that we're trying to use to get things specifically into cells to perhaps treat cancer and other disorders. So first of all, let's look at gap junctions. They're actually protein channels that will connect the two cells together. So their two cytoplasms are actually connected, and this allows passage of small molecules. Again, it's formed by transmembrane proteins that are going to form pores, and it's going to be very important for synchronizing function and cellular differentiation. As you see in this picture, you've got gap junctions formed between two cells here, and you've got the intercellular gap between the two cells, and this then will allow the molecules to move directly from one to the other, and you can see how important that would be if you want to synchronize heart muscle contractions. Here on the lower right is just more actually the way they would look, so but I don't want you to worry about this, but just get the idea of what the connections look like. Direct contact between two cells can occur with cell membrane receptors. The ligand is also going to be something on the surface of the other cell, or it could be something attached or a component of the extracellular matrix. Now these type of interactions are extremely important between lymphocytes and generating or developing an immune response, and they can also serve as scaffolds and help regulate cell growth and differentiation so that cells don't overgrow, that they stay in the right spot and differentiate properly. And to get an idea what this kind of contact or communication would look like, you see very simply here you would have the signaling cell that would have its ligand on the cell surface. The adjacent target cell would have receptors for it. If you want to look at cells attached to the extracellular matrix, so here's the nucleus. Here would be something just inside the cell membrane. This would go through the cell membrane, and then these fibers, these would be proteins that would attach to various components within the extracellular matrix. And that would, again, changes in the extracellular matrix would then trigger signals down into the nucleus. So this is via direct contact. The most common form of communication between two cells is secretion. In other words, the signaling cell will produce something and secrete it outside of the cell. One of the more common ones is paracrine signaling. These are chemicals that are produced in one cell and then are secreted just in a localized area, and they're rapidly destroyed. So their signal is limited to cells that are adjacent or near the signaling cell. So the target cells have to be near it. Autocrine signaling represents cells that respond to ligands that are secreted by the same cell. So the autocrine, the signaling cell, and the target cell are one and the same. This happens in development of the immune response, but it also happens in some tumor cells. Hormone or endocrine or neurohormonal, as your text refers to as signaling, this is where a hormone will travel through the bloodstream to target cells that are far apart from the cell that made the signal. And this is true for endocrine and even from neurohormonal signaling. But neurotransmitter or synaptic signaling, again, is a little bit more like paracrine, but it's so much closer, it is considered a different form. And so it's confined to cells of the nervous system, and of course, is going to occur at these junctions between nerve cells and the target cells. These type of secreted mechanisms of signaling are represented in this figure. So we have our contact dependent, our paracrine, the close, autocrine the same, hormonal distant, neurohormonal distant, neurotransmitter at a very close target cell. Exosomes are something that's pretty new. We haven't known about them for all that long. They're little cell-derived vesicles, which means that, of course, they're surrounded by phospholipid bilayer. We find them present in urine and blood and any of the other biological fluids. Their formation sounds kind of weird to start. So you have, say, an endosomal membrane. So let's talk about, let's say we have a lysosome or something, or phagosome. And it's inside the cell. So we're talking about a vesicle that's surrounded by a phospholipid. And within that vesicle, the membrane is going to invaginate, so move inward to form a lot of little smaller membrane-bound vesicles within this. Okay, those within the endosome that's within the cell are called exosomes. You say, what? Well, just wait. Anyway, so the exosomes then, since there's a lot of them in now what was this one endosome, is referred to as a multivesicular body, or MVP. Now, what happens is when the MVP fuses with the cytoplasmic membrane, the inner content of those little vesicles inside the exosomes are released outside the cell. Okay? And we find that these exosomes are important in coagulation, intercellular communication, obviously why we're talking about it, and also waste management for the cell. All right. So, picture of how this works. So let's go down here. And so let's say endocytosis occurs and we have an endosome, which is now bigger here. And what's happening is forming these little exosomes inside an endosome. Okay, so you get a whole bunch of these in here. When this then fuses with the plasma membrane, these things are released. These are the exosomes. So it's not easy to describe, but if you take a look at the picture, it kind of makes sense. Why am I telling you about this? Well, they're kind of unique. They're very small. Their lipid composition is different from what we normally see. And we have seen that they can contain proteins and RNA molecules that function in various target cells. So we think that we might be able to use them as cell-free cancer vaccines. In fact, there are actual trials going on right now in some patients that have tumors to see if we can boost the immune response by putting some of the cancer cells and stuff in these little exosomes and delivering them to the immune cells. So that's why I brought it up. Kind of cool, I think. Which brings us to a discussion of cell-to-cell adhesions. I apologize for how busy this slide is, but I didn't want to have a lot of slides, and you booked one into this and about a little bit more detail than I felt was really necessary. So I tried to combine it into just a few slides, which then became very busy. But anyway, plasma membranes hold groups of cells together, forming tissues and organs. And they do this because they're held together by extracellular matrix, cell adhesion molecules, or specialized cell junctions. The extracellular matrix itself, including basement membranes, is secreted by cells, mostly fibroblasts, and they are going to form this fibrous mesh or a glue that will hold the cells together. Now, one of the functions of the extracellular matrix is to provide a pathway for nutrients and waste and anything soluble between blood and the tissue cells. Within the matrix, we're going to find things like collagen and elastin, also some very large adhesive glycoproteins, fibronectin, more proteoglycans, and hyaluronic acid. If you get really involved in connective tissue, these are things that you need to go into in a little bit more depth. The basement membrane and basal lamina, these are thin layers of connective tissue underlying the epithelium of many organs, which is why it is something that there's a lot of in the human body. Macromolecules are found the extracellular are found in intercellular junctions and at cell surfaces. And the ECM, or the extracellular matrix, can form an interstitial matrix, again, or basement membrane. We've already mentioned it's secreted by fibroblasts. And of course, those form the connective tissue, which connects cells together to form tissue and organs. Your book shows a lot of pictures of the different types of human connective tissue. This should be stuff that you are somewhat familiar with, so I'm going to kind of gloss over it here. Mentioning, of course, you can have bone, which is hard and dense, and tendons and skin, which are flexible, and cartilage, which is shock absorbing. And however, the extracellular matrix is also very important for regulating cell growth, movement, and differentiation, and development too from a fertilized egg, which we all came from. This slide again is just a figure of the extracellular matrix, the different forms. So you can have the fibroblasts that make it, and you have basement membrane here, different types of collagen, interstitial matrix here in a tissue, just to give you a sense of how important these extracellular matrices are. Which gets us, of course, to talking about cell adhesion molecules, or CAM. And these are very important. They're important when we talk about the formation of tissues and stuff, but also for activation of cells, cell migration, and a number of things. So I'm going to spend a little time with the cell adhesion molecules. They bind cells to adjacent cells, again, components of the extracellular matrix. And this is something that's going to be useful for you to know, these 4 protein families of the CAMs, or cell adhesion molecules. Integrins, which binds to the extracellular matrix. Cadherins, they got their name because they're calcium dependent, but they're found, they have a unique tissue pattern. They're found mostly in epithelial cells. Selectins bind to carbohydrates, and they're very useful and important in some migration of cells. And then we have a very large family of CAMs, or cell adhesion molecules, known as the immunoglobulin superfamily of CAMs, which is abbreviated IGSF CAMs. Anyway, they bind to other CAMs, okay, and other immunoglobulin CAMs. So these are the four major protein families of CAMs, and can be very important for a number of different processes within the cell. Now there are some specialized cell junctions. They can involve direct physical contact. They will again hold cell togethers or allow small molecules to pass. There are three types of these specialized cell junctions. Desmosomes, which form kind of buttons, and they act as a brace. Tight junctions, like you see in the blood-brain barrier, prevent movement, and then of course we've already talked about gap junctions. And again, we have a nice little final picture here summarizing some of this. So we have some of our desmosomes and a tight junction here, and the gap junctions with the gaps, but I think the other picture was a little bit better. So this is just, again, getting us a more in-depth discussion of the various proteins that are important for cell functioning and holding the cells together to form tissues and organs. We will, in the next lecture, then go on to talk about metabolism, which is also very important, and then we will move on into what happens when cells sustained injury. So talk to you soon.